So, Laith, over to you.
Thank you so much.
Thank you.
It's Laith Alexander. As Sarah so nicely introduced, I'm an academic clinical fellow in psychiatry at King's College London. And I'm really delighted to be giving this talk today about the neuroanatomy of depression. It's a little bit didactic, but hopefully it's a broad overview, and some of the anatomy, well, all of the anatomy is particularly relevant to how ketamine works. The image on the right of the slide is Dali's artistic interpretation of the neurobiology of depression. And I'm not really sure there's like a half orange in the interpretation, so I'm not quite sure what that is, but everything else kind of checks out, I guess. Just to give you a brief overview of what I'll be talking about, I'm going to focus on some of the historical perspectives to start with, and then cover the structural abnormalities that have been identified in countless neuroimaging studies relevant to depression, then focusing on functional abnormalities, brief, I guess, circuit perspective, looking at the default mode network, and then bringing a focus on ketamine and what we've learned about the neurobiology of depression from ketamine's mechanism of action, and then just some concluding statements. So when I was asked to give this talk, I think the suggested title was a revised anatomy of melancholy. So it would be remiss of me not to talk about the historical perspectives and mention Robert Burton's famous work, which many of you are probably aware of. The Anatomy of Melancholy was published in 1621 by Robert Burton at the age of 43, and he wrote under his pen name Democritus Jr. He had long suffered from what was probably depression, which extended his tutelage at the University of Oxford. So initially I think he was at Brasenose and then moved to Christchurch. A rumour that was perpetuated at the time by one of his colleagues, which is perhaps referenced in the middle quote on this slide, was that his melancholia was caused by him reading an astrological chart that predicted his death in 1640. And his death was probably actually falsely rumoured to be death by suicide. Whilst melancholy may or may not have killed Burton, it clearly occupied much of his life. And the anatomy of melancholy was partly an exercise in him keeping himself busy. William Osler, the famous Canadian physician, said that Burton's book was the greatest medical treatise ever written by a layman. So what exactly did his book anatomize? Well, at the time, melancholy was defined actually as a subtype of delirium or mental distress, and it involved three things: impairment of the mental faculties, predominantly reason and imagination, passions of the fear and sorrow, and also, rather interestingly, an absence of fever. The etiology at the time was explained in terms of four humours, Galen's four humours, in this case an excess of black bile, which was described as cold and dry. And the excess of black bile affects both mind and body, so really instantiating melancholy as both a disease of the mind but also of the body. And I think rather importantly and relevant to sort of biopsychosocial formulations we have in psychiatry today, Burton emphasised the importance of societal factors that produce melancholy. So it wasn't just a private state to the individual, but actually Burton believed that it was an epidemic, a Europe-wide epidemic, in the context of pronounced religious war and political upheaval at the time. And in the second half of his book, or the second third, sorry, he covers various forms of melancholy, including the misery of scholars, which I think is rather relevant. He also touches on treatments, several of which are known to be effective today. So, for example, getting out, fresh air, exercise. And his book is interesting in so much that the text was actually written to be therapeutic itself, and not just a summary of case histories, which was more common at the time. And on the right, on the slide here, you can see Burton's bust on his tomb in Christchurch College, and the Latin is roughly translated as "melancholy gave me life and death." We have come a long way since Galen's humoral theory, which predominated at the time of Burton writing this book. And since the latter half of the 20th century, multiple different explanatory frameworks have been developed to try and understand the etiology and pathogenesis of depression. So we have, perhaps most prominently, neurochemical theories, which focus on initially monoamines, now glutamate, GABA, and other neurotransmitter systems. We also have neuroendocrine and neuroimmune hypotheses, which focus on dysfunction in the HPA axis, neuroanatomical accounts, which are both structural and functional. And then also we have cognitive accounts, which focus on abnormalities of hot cognition, which are, for example, negative affective biases, impairments in motivation and reward processing, and also cold cognition, which focus on working memory and attention. In this talk, I'm going to focus predominantly on the neuroanatomical level of analysis. We know that there are structural and functional abnormalities in the brains of people with depression and major depressive disorders, and I'm going to turn to structural abnormalities first, and then review some of the more consistent functional findings. So structural abnormalities in the brain have been found in both grey and white matter, and in this talk, because it's quite a broad overview, I want to focus predominantly on meta-analytic evidence implicating different structures in the brain in depression. So in the grey matter, abnormalities have been reported in the prefrontal cortex, the hippocampus, and structures within the basal ganglia, and typically lateral ventricular volumes are also increased. So you can see on the right-hand side of the slide here, this is taken from a paper in 2011, just reviewing several studies that looked at structural MRI changes in the brains of depressed people. And you can see that at the top, there's increases in lateral ventricular volume, and then there are reduction in, so orange are significant changes, the reduction in volumes in several prefrontal areas, including the orbitofrontal cortex, the caudate, the putamen, and also the hippocampus. Why these structural changes happen isn't entirely clear. It may be related to increased neuronal cell death or glial cell death. It may be related to impairments in adult neurogenesis, for example, in the hippocampus. And rather interestingly, it may also reflect damage to dendritic arborization on neurons, for example, in the prefrontal cortex and the hippocampus, which are particularly sensitive to the effects of chronic stress. And through these accounts, I'm going to highlight some specific brain regions that are relevant. So here I just want to talk briefly about the hippocampus, which I'm hopefully not teaching you to suck eggs, but just briefly, it sits in the medial temporal lobe. It's a critical structure in episodic memory, in contextual fear conditioning, which we predominantly know from rodent studies, and also in memory consolidation and reconsolidation. And the hippocampus, as I mentioned, is particularly sensitive to the effects of stress. And as the severity, which means both the intensity and the duration of stress increases, alterations in hippocampal neurogenesis, plasticity, cytoarchitecture, and activity occur, which are predominantly initially facilitative and probably help the person, the individual, deal with stress. But obviously, if this is prolonged over time, it can eventually degrade cognitive and psychological functions that are mediated by the hippocampus. And the change in cell architecture, as mentioned, might account for some of the shrinkage in hippocampal volumes. The hippocampus sits at the center of a network of structures that are important in signaling aspects of stress. So the arousal and excitability associated with stress is mediated predominantly through the HPA axis, and the perceived aversiveness, possibly through the amygdala. That's possibly slightly simplistic accounts. But these contribute to the effects of stress, both good and bad, on the hippocampus. And then projections from the medial prefrontal cortex, which I'll touch on again a bit later, probably signal the controllability of stress, partly through connections to the dorsal raphe system. So moving on briefly to white matter changes that are seen in structural scans associated with depression. White matter lesions are commonly observed, and they're particularly predominant in later life depression, thought to be ischemic, and an increased burden of white matter lesions are associated with a greater symptom burden, worse prognosis, and particular symptom domains, so for example, apathy and psychomotor slowing. So sometimes this type of depression is thought to kind of reflect its own subtype. And damage to connections between the frontal cortex and limbic regions might underlie this type of depression. And on the right are just several examples of MRI scans from patients who've suffered depression over the age of 65 and show a degree of cognitive impairment together with white matter hyperintensities, which are, I guess, rather unhelpfully shaded red, but they're there. Now moving on from structural changes to functional changes associated with depression. There are countless functional neuroimaging studies exploring changes associated with major depressive disorder and the vulnerability to it. However, the replicability in functional neuroimaging studies is notoriously challenging. The costs associated with these studies often mean the sample sizes are small. There are different methods and modalities used when I talk about functional neuroimaging. This could include fMRI, PET, and EEG, which don't always generalize to one another. And there are also issues with interpretations of results. So, for example, imaging changes are correlative. We don't know if any of the regions are causally involved. That's why preclinical studies in animals are particularly important. With the advent of meta-analytic approaches that synthesize a vast amount of neuroimaging data, we can glean some insights into consistent changes across many studies, although obviously the meta-analyses are only as good as the individual studies themselves. Just touching on meta-analysis of emotion activation studies to start with. So these are studies in healthy volunteers which give us insight into the brain regions associated with the induction of certain emotions. And I guess particularly relevant here is non-pathological sad emotion. This paper is old. It was, 2002, which summarizes 55 PET and fMRI studies. And it looked at consistent activations across the brain associated with different emotions. They divided the brain in this study into 20 non-overlapping regions and basically counted the number of studies that showed activations in those brain regions and did simple statistics to compare and contrast activations with different emotions in the different brain regions. And with sadness, the most robust finding was activation of the subgenual anterior cingulate cortex, which was focused on Brodmann area 25, which is the one cytoarchitectonic subdivision which constitutes that brain region. Only 46% of studies that they summarised actually found this, and they interpreted that as differences in scanning methods. So the studies that most consistently showed activation of this brain region imaged people once the desired emotional state was actually achieved rather than during the induction of emotion. So the anterior cingulate cortex, which is shown here, sits on the medial surface of the prefrontal cortex, and it's typically divided into three subdivisions: the dorsal ACC, which you can see at the top, the perigenual ACC, which sits around the genu of the corpus callosum, and then the subgenual ACC, beneath the genu. So genu in Latin means knee, because the corpus callosum kind of looks like a knee. And different Brodmann areas, which are numbered in this diagram, constitute each division. And you often hear about, and I've already talked about, area 25 being implicated in depression and low mood. And this actually sits right at the back, right at the caudal area of the subgenual, which is highlighted in blue. The subgenual ACC has been implicated in countless studies, imaging normal sadness. Also, it's shown activation in other emotional states like happiness, and also in pathological states of low mood, and perhaps most famously, Helen Mayberg's work is relevant here. The subgenual ACC was obviously subsequently targeted in deep brain stimulations studies, looking at the use of this technology to treat depression, which have shown mixed results so far. And this brain region is particularly interesting because its anatomy suggests that it's at an important juncture between cognition, emotion, and also autonomic function. And we've gained many insights from track tracing studies, particularly the work of Helen Barbas in the States, in primates, and also from diffusion-weighted imaging studies in humans. It projects strongly and receives projections from higher prefrontal regions that are involved in executive function. So in particular, let me see if I can move my cursor. Is that working? Yeah. So this is macaque brain, a medial view of the macaque brain. And you can see projections from and to higher prefrontal regions, which include area 10, which is frontopolar cortex, and area 14, which is the rostral part of the orbitofrontal cortex, and also receives projections to and from the dorsolateral prefrontal cortex. These regions are commonly involved and postulated to be in the top-down regulation of low mood. The area 25 also projects strongly via the unsonate fasciculus, which you can see in this diffusion-weighted imaging track tracing study in humans, to structures in the medial temporal lobe, such as the amygdala and the hippocampus and the perirhinal cortex. And this is implicated, obviously, in memory and may be important in the maladaptive rumination associated with depression. Frontostriatal fibers, which you can see, slightly difficult to see, but labelled in dark blue there, are predominantly projecting to regions of the ventral striatum, so the accumbens, ventral caudate, and ventral putamen, which might be particularly important in reward-related symptoms of depression, like anhedonia. And then also, I think, rather interestingly, the area 25 is quite unique in that it projects to, you can't actually really see it here, but it projects to brainstem regions and the hypothalamus, which are critically involved in endocrine and autonomic regulation. And of all prefrontal regions, area 25 seems to have the strongest projections to regions like the hypothalamus and amygdala. Activity changes within the ACC, both the subgenual but also the other divisions that I talked about, have been implicated in the successful treatment of depression via a variety of means. This includes psychological therapy, pharmacological therapy, and also deep brain stimulation. You can see actually in the top right here, pre-treatment activity within the perigenual ACC has putatively been identified as a biomarker which could predict depression recovery, although variable replication of this. There are several more, novel approaches looking at meta-analyses of neuroimaging studies. So, for example, many of you will have heard of neurosynth, which uses an automated method to extract coordinates from thousands and thousands of different studies. And then it compares the frequency of coordinates reported for search terms versus those that don't contain search terms. And then a map of coordinates are generated where papers which show coordinates that are significantly more frequently activated for those search terms are highlighted compared to those that don't. And these meta-analytic approaches are very useful, but they have their limits. There's a substantial lumping of data, especially with patient populations, so youth and adults, people who are medicated and not medicated, and also both resting state and emotional tasks and cognitive tasks are included. But you can see here that from the neurosynth search of just depression and anxiety, it highlights the amygdala and the subgenual ACC as being implicated. Briefly touching on the amygdala and its role in depression, it's been rather extensively, as I'm sure you know, implicated in depression and anxiety disorders. And the amygdala is an almond-shaped nucleus that sits just in front of the hippocampus. And broadly, amygdala hyperactivity, particularly left amygdala hyperactivity, is thought to increase the risk of developing depression and other internalizing illnesses such as anxiety. And evidence comes from several sources, both heightened amygdala reactivity to emotional sphases, which is associated with the subsequent risk of developing internalizing illness, treatment of SSRIs damping amygdala responses to emotional phases, and also coordinate-based meta-analysis, like the one shown in the top right here, which identify left amygdala hyperactivity in people with depression. But the evidence for amygdala reactivity in depression is very inconsistent. I think this study was done in Oxford, partly in Oxford, in a large study of over 20,000 people from the UK Biobank. Tam and colleagues, they explored the relationship between amygdala activity and self-reported depression scores across the cohort, and they actually found no association. There was a very small correlation between the degree of amygdala reactivity and a stricter clinical criteria for major depression, but the strength of association was extremely small, a Cohen's d of 0.03. So is all lost? Well, I think it suggests that probably simple box and arrow explanations highlighting and focusing on single brain regions probably are not sufficient, and that still we need to develop better biomarkers of depressive symptoms. I just briefly wanted to touch on the default mode network in depression. This is partly because it's, I guess, a relatively more novel circuit-based perspective that has emerged over the past 15 or 20 years. And the default mode network is a network of brain regions, including the rostral ventromedial prefrontal cortex, which is abbreviated RVMPFC there, and the posterior cingulate cortex, precuneus, and medial temporal lobe. And these regions are typically more activated in states of rest and deactivated during task conditions. Given the role of the default mode network in autobiographical and self-referential thinking, it's perhaps no surprise that this network of brain regions has been linked to the ruminative thinking which dominates in depression. And increased connectivity between the subgenual ACC, which I mentioned earlier, involves in negative emotion, which is highlighted in the diagram on the right here in grey. And nodes of the default mode network have been postulated to underlie the negative, affect-laden, behaviourally withdrawn, ruminative thinking that's seen in depression. And interestingly, ketamine has been shown in a small number of studies to reduce functional connectivity between the subgenual and key nodes of the default mode network, for example, the perigenual ACC, which is part of the rostral ventromedial prefrontal cortex, and the posterior cingulate cortex. So in the next section, I just very broad brushstrokes wanted to touch on what we've learned from ketamine to make it particularly relevant to this conference about the neurobiology of depression. And I want to draw sort of very broad themes for the sake of brevity, and given that the literature is obviously quite complicated. Firstly, we know that ketamine seems to induce a surge of glutamate levels within the prefrontal cortex acutely, including the medial PSC and anterior cingulate cortex. This glutamate surge is thought to be mediated by ketamine's antagonistic effects on NMDA receptors, on inhibitory interneurons, which may remove the brake in excitatory neurotransmission. Or alternatively, it might be mediated through ketamine's effects on opioid receptors, which we'll hear in Luke's talk later. And the surge in glutamate likely induces neuroplastic changes over the course of hours to days. Secondly, projections from the medial prefrontal cortex and the anterior cingulate cortex to the hippocampus have been causally implicated from rodent studies in ketamine's antidepressant effects. So in this study in 2016, which is in the middle here, they used optogenetic strategies to specifically activate projections from the medial PFC to the ventral hippocampus, and they found that this could recapitulate ketamine's rapid antidepressant-like effects on the force-swim test in rodents. Third, projections from the anterior cingulate cortex to the ventral striatum, which, as mentioned, includes the accumbens, have been implicated particularly in ketamine's anti-anhedonic effects and effects to ameliorate reward processing symptoms in depression. And obviously, a fourth thing, which I've not mentioned here but touched on previously, was the effects of ketamine to modulate connectivity within the default mode network and between regions like the subgenual and the default mode network to alleviate perhaps ruminative symptoms in depression. One region that's, gosh, it feels quite nerve-wracking talking about the habenula in front of Professor Hu. One region that has been received a lot of focus, particularly in the context of ketamine's antidepressant effect, is the lateral habenula. So the habenula is part of the epithalamus, so it sits just above the thalamus, and it's an evolutionarily ancient brain structure present in almost all vertebrates. It plays an important role in behavioural choice, and in particular, as summarised in this paper in 2010, seems to inhibit motor outputs in response to the anticipation of aversive outcomes and in response to pain and stress. It does this partly through the inhibition of brainstem neurotransmitter systems. And numerous lines of evidence implicate the lateral habenula in major depression in humans. Its connectivity places it at the junction between mood, motor function, and monoaminergic systems, together with the regulation of the sleep-wake cycle. And for example, there are several studies that have implicated a change in activity in depression in the lateral habenula, but for example, it's been shown that plasma tryptophan depletion in people who have a history and current depression seems to activate the lateral habenula. And the work of Professor Hu and yes, hopefully I'm doing justice to your work. The work of Professor Hu and colleagues has provided really important insights into how ketamine acts on the lateral habenula to induce its antidepressant effects. In rats, they've shown that ketamine seems to block burst firing in the lateral habenula associated with chronic restraint stress, and shown this is shown left, and that perhaps through blocking the burst firing in the lateral habenula, this could revive reward centres in the brain to elevate mood. More recently, the group has shown that ketamine seems to be trapped in the NMDA receptor channel pore, which might explain its longer-lasting effects after several times after its half-life, after it's eliminated in the body. And shown on the right here, using rodent lateral habenula slices, they bathed these slices in ketamine and then subsequently washed out the ketamine. And despite the washout, you can see in black with no stimulus, the effect on lateral habenula burst firing remains. However, when there was paired presynaptic and postsynaptic stimulation in these slices, putatively therefore opening the NMDA receptor channel pores and therefore kicking out the ketamine that was trapped there, they found that this change in the NMDA receptor-related excitatory postsynaptic potentials diminished, which is what's used to support the evidence that ketamine's putatively trapped in channel pores in the habenula. So that was a bit of a whistle-stop tour. And as you can probably tell from that, we, I guess, paradoxically know a lot, but also a little about the neuroanatomy of depression. And there are plenty of known knowns, but there are also plenty of known unknowns and possibly unknown unknowns. We haven't reached the overall goal, I would say, of a comprehensive, integrated neurobiological account of depression and an account that accounts for these different levels of analysis. There are different perspectives, corticolimbic perspectives, corticostriatal, network perspectives on the default mode network, which kind of still exist in parallel. Why aren't things clearer? I suppose there are multiple reasons. These are just a few. Depression is very heterogeneous, and the etiology and pathogenesis is probably very different in different people. Particularly, I guess, relevant to this talk, where I've talked a lot about neuroimaging, studies in humans can rarely show causality. I suppose lesion studies are the really kind of only instance of that. And while studies in animals can demonstrate causality in rodents and primates, there are issues with translational validity. So, for example, using things like the forced swim test as a model of depressogenic behavior. And then neuroimaging studies, as I've touched on earlier in the talk, have limited reliability, and that's predominantly due to the small sample sizes. So where next? I suppose there's questions about whether individual symptoms can be linked to specific brain regions or more likely networks of brain regions. For example, could cognitive abnormalities be linked to abnormalities in the dorsolateral or dorsal prefrontal cortex? Could diminished reward processing be particularly linked to connections between the prefrontal cortex and ventral striatum? As I touched on in the previous slide, different accounts of depression, neuroanatomical, neurochemical, neuroendocrine, will need to be integrated into more parsimonious accounts. And there may even be levels of explanation that are yet to be discovered or only recently being discovered. So, for example, neuroimmune level of explanation, and also the gut microbiome is perhaps another good example of that. And I think coming back to Robert Burton's work, the heterogeneity in depression was appreciated in his book. I think it's almost as long as Les Miserables. It's a very, very long piece of work, over 500,000 words. And that's because he touches on so many different causes of melancholy. And we've always understood that depression is not unitary, but sometimes I think this appreciation can get lost when trying to draw generalities from research. And going forward, robust clinical phenotyping and an appreciation of individual differences in depression will no doubt help us elucidate the neural mechanisms underpinning this common and important disease. So thank you so much for listening. Hope that was useful. These are the references which you can ask me for. Thank you.
Thank you, Laith. Terrific overview. We may have time for one or two questions. Please.
Thank you for the talk. I was fascinated by your discussion of the role of the habenula in depression and ketamine's potential role there. Have any other psychedelic drugs been examined in kind of their potential antidepressant role, like psilocybin, and what effect it has on the habenula as well?
That's when I was reading the paper that was published in 2023 showing the blockage of the pore, I was thinking the same thing, and I don't know, Professor, there are.
So these two people from my lab are working on neurosynth on lateral habenula. So hopefully they will tell you the answer maybe next year.
Yeah, go ahead.
We use mouse models. Would rat models be better?
We use both in rodent studies, both mice and rats. The forced swim test is sometimes done in rats as well. I don't think it's a question, I think it's more a question of actually the construct, face, validity of and predictive validity of the different models. Almost regardless of what species they're in, if that makes sense, the model is more important. So in primates, we use perhaps slightly more sophisticated models because we can model depression-like behaviors a bit better. I think certainly things like forced swim test and tail suspension tests have a degree of face validity perhaps, and have very good predictive validity, generally speaking. And I suppose they're really used as a screen for potential antidepressant activity of various different drugs. But the construct validity is called into question. And so therefore, when we're making conclusions about the biology of depression in humans from these tests, I think we have to be very mindful of that and recognize that actually probably the circuits that underlie, for example, a rodent swimming or not swimming in a bath of water are very different from what underlies the depressive syndrome in humans. So, for example, I don't know, you could interpret increased mobility in the forced swim test as increased anxiety, but increased mobility is typically used to suggest an antidepressant-like effect, if that makes sense. And we know depression and anxiety are highly comorbid, so that doesn't quite match up. So I think it's just being mindful of the limitations in terms of validity when we use the tests. Yeah.
I think we have time for one last question, Robert.
Thank you for an interesting and concise overview of a complicated topic. In the end, you say there's 3 venues towards becoming more precise. I was wondering, what would you think is a promising approach to clinical phenotyping?
That's a very good question. I think, in my opinion, part of it relies on age-old techniques of careful history taking and being very, very clear which symptoms, which symptom domains are present in different populations. I think the problem is when we use scales, like, for example, the MADRS or something of that sort, we lump together many different symptoms into one number, and we lose that appreciation of heterogeneity there. And I think there are limitations by and large in the questions themselves, and they don't always ask exactly what are relevant to patients. So I think that's part of it. I also think the advent of things like patient-reported outcome measures is probably going to be very useful. So, for example, take social functioning. So there's like SFQ, social functioning questionnaires. Those are outcomes that really, really matter to patients and are probably, and well, definitely affected in things like depression. And perhaps by focusing on those things, we might get a bit of a better account of the underlying neurobiology. And that's relevant to animal studies as well. So, for example, measuring social function in non-human primates, looking at social function in rodents. All these things can be, we need to think carefully, but they can be translationally explored. I don't know if that makes sense. But yeah, that was my opinion. Yeah, that's okay.
All right, thank you very much.
Yeah,
I think
thank
this
you.
concludes our morning.
Thank you. Thanks.
And we are now heading towards the lunch break, and we'll be back at 12:40. See you soon.
Thank you. Thank you.